So welcome everyone. This is lecture 11 of engineering 443 computational biology modeling methods with virtual tissues. We will be talking today about how to use CC3D Python API. We will come back to anti-money models where we left off last week, and we will use the delta notch example to introduce the idea of staging panels, which you would need for deployment of your projects in NanoHub. We will review as we do. Just a reminder: the class is being recorded and streamed in YouTube. We will review the basic ideas of subcellular modeling using SBML that we've been discussing the last two lectures. And after this review, I will pass on to TJ, who will be presenting about how to use CC3D Python API. And once we're cover that material, which is essential, if we have time, we'll come back to the delta notch model that we were working with last week and run the base model, and then include cell growth and division on top of this current cell model to see how we can use subcellular components to control what's happening in the cellular pods implementation. And then we'll use this example to introduce the idea of staging panels. So I will pass on to James, who's gonna go out of the room checking on students' updates with their projects.
Okay, so maybe just start at the top. Devin, do you want to give us a quick update?
Yeah, did you want me to share?
We can, Josh, if you want to unshare, then we can.
Yeah, I just made a couple slides here.
I mean, in principle, first versions of projects are due in a week.
Yeah.
So I know
so
that's
yeah. So this is showing my tumor project and how over time the cancer begins to stratify and spread out. So within a month of the beginning tumor, and then a year, you can see it begins to branch out. And then I also have the field to show the damage accumulation due to glucose starvation and pH. So you can see that the outer cells, which are newer, don't have accumulated damage yet, but the inside ones that are becoming starved do have some. And that's about all on that. And then to present my data better for kind of the presentation, I want to make plots of each of the types of cell count over time for like the quiescent, proliferative, and necrotic cells, stem and cancer cells, and then do parameter scans to test varying parameters. And then I don't know if I'll have it done for next week, but I want to try to apply tracking to see when mutations occur, just by knowing that all of the tumor cells start out as quiescent stem cells, maybe just track how long it takes for them to move on to a different type, and then upload to NanoHub. And that's all.
great. The biggest thing with tracking is it's easy enough to say whatever cell divides, you put onto a stack the identity of the parent. So you have the lineage. What's a little bit more work is figuring out what to do with that information. And so typically in experimental, when they do this in experiments, what they'll do is they'll color the cells that say all of these cells are descended from cell such and such, all of these cells are colored descended from such and such. But you have to decide at what time you're going to decide that. And sometimes they'll also do a histogram where they'll say, of the cells at the end of the simulation, at time X in the simulation, half of them are descended from this cell, 1/4 of them are descended from this cell, 5% are descended from this cell, and so on. But what the way that the picture appears and the way those histograms work depends on the time at which you start. After all of the cells are descended. If you start with one cell, they're all descended from one cell. And so you have to say, okay, if there's a mutation at time X, then you have to ask the question, which cells are descended from in that lineage? And so you need then to write something that lets the user change the reference time or the reference cell for the lineage tracking. And that takes a little bit of thought. We could probably dig it out. Priyam had a lecture for our CompuCell course a couple of years ago where she showed how to do that. And there, if you look in the literature, there's sort of standard ways of thinking about that display. The tumor winds up essentially looking like a pie, which is chopped into wedges of different widths, it's actually quite interesting to see how the fraction of cells of a given lineage change in time, because you could have a situation where there's a mutation that which sweeps, where every cell becomes descended from that. There are others where you get a lot of cells descended from a cell, then they all disappear. There are others where you have competing lineages, which are more or less the same. But you might want to look up to how to display effective ways to display lineages. And if not, one place you see that a lot is when you're doing bacterial colony growth. They have two-dimensional colonies that grow outwards, a little bit like a tumor. And so the way they color things to display that is not a bad thing to look at. So there's some pretty good literature on that. Again, once you have the stack of ancestors, it's not so bad. But you do that, have to write some code to walk the various lists and put it together. If you don't care about it being fast, it's not so bad. If you want it to be efficient, then creating the trees is a little bit of work. Andy might, I don't want to talk about it right now, but Andy might have some ideas and ways to help you with the tree walking for that. That's something he's good at. But that's great. I'm pleased. Thank you. Okay, Kali, I guess you're next.
Sorry, I was muted there. Let's do this presentation. All right, so this PowerPoint doesn't have all of my update, but it has a big part of my update. So one of my big goals for this week was debugging my cyclic AMP model for D-Discordium. The problem, if you remember from last week, was that we weren't observing oscillatory behavior in the previous implementation, like the level of cyclic AMP just kept increasing. We weren't seeing kind of a wave-like pattern. So the solution is with Dr. Glazier, I rebuilt the model from the ground up, starting with the baseline model that I was able to replicate from an earlier paper in Tellurium. So here's the result of that baseline implementation, coupling with the external and coupling the external cyclic AMP field with the internal cyclic AMP levels. And as you can see here, we seeded 4 oscillatory cells in the center of the domain. So the cyclic AMP signal is radiating outward from that center. So the level of cyclic AMP, you can't really see just from one screenshot, but the level of cyclic AMP in the oscillatory center will rise and fall, leading to these wave patterns, and the signal is relayed by the rest of the cells, which are all relay cells. So that's good. We have a proper implementation now. The next step of that, which I worked on following that meeting, was now adding back in ammonia interference to the cyclic AMP model. If you recall, ammonia is supposed to inhibit cyclic AMP dynamics in this model. So the paper that I'm using is a bit vague. on ammonia interference. It's kind of unclear whether it's based on the amount of ammonia or the concentration of ammonia. So trying both methods, if you look at by amount, both of these screenshots are taken after around 300 Monte Carlo steps. So if we look at by amount, we have the cyclic AMP concentration, external concentration, significantly decreased, like inhibited, you could say, as opposed to what we saw previously. And the internal cyclic AMP, at the very beginning, there's a little spike in the center around the oscillatory cells, but then eventually that will dissipate. So we're seeing inhibitory behavior, but it's probably a bit too much. So now trying the method based on concentration, we're seeing a similar pattern as we saw earlier. And it's difficult to see in the photo, but the maximum amount of cyclic AMP is pretty similar to what it was before. So we're kind of in this interesting conundrum where like one option that kind of gets rid of the signal altogether and another option in which the impact of ammonia is negligible. So, I mean, I don't know if there's a third option, but right now my gut instinct is to say that it's better to choose a model with the wave behavior than without it. It could also be possible that the N that I think is ammonia concentration in the model might be just some other factor. Again, the wording in the paper is kind of weird. I don't know what the implication is that the ammonia interference could be negligible. At that point, if it's negligible, like, do I still put it in the model? Do I leave it out if it's basically the same? I mean, that would definitely probably save some computational time. So I would love some advice on that if anyone has it. Now, if anyone wants to see the NanoHub, I could go there as well.
Yeah, if you've got it running, it would be great. It's nice to see the waves in action.
Yeah.
I mean, one thing I might try before you get too involved in the ammonia. The ammonia is interesting because it leads to modulation of the oscillation. It changes the frequency of the oscillation, but it's not a huge effect. But one thing is that you get waves even when the cell's density isn't filling all the space. And so one thing you might want to do is instead of having your cells fill all the space confluently, have gaps between them and ask the question, do you still get traveling waves? And adjust your parameters so you get waves even if the cells are a few cell diameters apart.
Right.
And if you have that, then you can have them chemotax. And then you should be able to see everything. And then again, the ammonia effect doesn't have to be very big. It's actually pretty subtle. It changes the way the direction of the aggregate moves.
Right.
But it doesn't abolish the oscillations.
Got it. It
And
could
so
also
one
be
thing
that there's enough, the ammonia has a greater effect when we have more cells, like in the actual model that we just
Sure.
had before.
Because they're going to be producing, right? They're more. It's going to accumulate more. Absolutely. The other thing I think that's important, and Josh and I were talking about this, is that we didn't really. Last semester, we built from scratch in class a bunch of models together. We took a paper and analyzed it, and this semester we didn't do that, partly because the models are a little more complicated. And so this approach that I think is something we should probably, if we teach it, when you get the class evaluations, you'll see I added some questions about some of these issues because I do want your feedback so we can do better next time. But I think that especially for these very complicated models, and a lot of people are building them, it's really important to start with the ODE model in tellurium, make sure it works. And then you take it and you put it in a single frozen cell in CompuCell, and you make sure it still works with different inputs and outputs as needed to make it work, to show what it does. Then maybe take two cells and put them next to each other and make sure that it still works. And then have those cells secrete into the chemical field with no diffusion and see what happens. If you're going to pull chemical out of the diffusion field, it's really important to know how much of it, whether it's concentration or amount, what the numerical value you're pulling has to be to make it work. And the easiest way to figure that out is using the tellurium, because it's fast. And then if you know that the value has to be 10 to the minus 11th, and you're reading the chemical field is returning 10 to the minus 15th, well, you're going to have to multiply 10 to the 4th. It's returning 10 to the minus 9th, you have to divide by 100. And so having a sense of what the parameter, what the ranges have to be to make it work is really useful. And doing that before things start moving around is even more useful, because once things start moving, you don't know whether the value is too low because you're in the wrong place or because the value is too low everywhere. And so I think this approach to systematically building more complex models is important. And I next time I'll do more on how to do that. We need the pieces though. The problem is we need to get the pieces before we could do that. And so it's a little bit of a bootstrapping problem. But the one you're doing here is a really great example. If you've got the NanoHub running, it would be great to see it.
Yeah, I have the NanoHub up. Let's see, do you want to see the baseline model or with ammonia?
Just looking for the baseline model first, since people, it's fun to see the.
Let me just change the string real quick. Okay. It takes a second before it loads. Yeah, so you have these 4 oscillatory cells in the middle and the rest are all relay. Okay, I'm gonna make a new window. Cyclic AMP. Okay, so the plot on the left is the external cyclic AMP field. The plot on the right is the internal field that is coupled to the model string. So we have internal cyclic AMP levels increasing the oscillatory cells. It's going to spread to the relay cells around it. Eventually, we're going to start to see that ring formation as the concentration of cyclic AMP in the oscillatory cells goes down again. Yeah, which happens right now. So the oscillatory concentration went down, but we're still going to see that signal being propagated. There we see a reemergence of that signal, second signal in the oscillatory cells. And the same thing's going to happen. Dr. Glazier, for the sake of my NanoHub app, I know Dr. Sego told me that having all these different trackers kind of takes up computational time. So which sort of trackers, if any, do you think would be the most relevant for my app?
Well, I think this internal cyclic AMP is very nice. That shows beautifully. The relay is the refractoriness is also nice. I don't know how much time it takes up. Do you have it multi-threaded? Did you try asking for 4 cores when you run it?
No, I don't think so.
So if we go to XML, at the very beginning, very top. Oh, you know, we are on 4 cores. Okay.
Yeah.
Okay, so that should be, there was one other thing so that's about is probably, I don't know, I don't know whether if you ask for 8 cores or 16 cores, it causes a problem or not. I don't have a tried. I did have one favor I wanted to ask you. If we could go into the XML down to the bottom where you create the initial condition. And I wondered if we could add just a couple of oscillatory cells at a different location. So if you copy line 168 to 174 and paste them in just below 174. And let's think we could sort of put them up in the corner. So something like start at, it's 256 by 256 lattice.
Mhm.
So let's start at something like 225. 225. We just need one maybe. So make it max would be say 230 by 230, 230. Because it's actually sort of interesting, and then save that one and run it. It's sort of interesting to see what happens if you have 2 waves collide with each other. Let's see where we are with it. Yeah. So that's very nice. Let's see if you're willing to try running that. It's sort of a fun. If I remember this right, it's sort of a fun. So if I don't, we'll go on.
Oh, should I have done like Newman boundary conditions instead of periodic? Well, I guess
No.
it doesn't really matter in this case.
Periodic is fine. We'll see what happens when the waves collide. What do they do to each other? Anybody have a guess what's going to happen when the waves hit each other?
I can't predict whether we're going to have constructive or destructive interference.
That's why I wanted to show it. See what happens.
Destructive?
And you get, again, it sometimes you have to run it for a while because it takes a few cycles, but you get what's called spiral wave chaos. And that, the particular parameters we have, it doesn't look like any of the relay cells are spontaneously oscillating. If you tune it, the excitability so that the amount you pull out of the chemical field is just right, you can get a situation where even the non-oscillating cells will oscillate because the field will come back and re-stimulate them. And you could get secondary oscillation centers, even though they're not intrinsically oscillatory cells. And when that happens, all bets are off about the pattern. You can get quite.
Is that excitability value something I should seek to find, or was that a fun fact?
Well, it's interesting to find it. I know this model does it, and so the question is, again, that for the purposes of if you want to do aggregation, it may not be essential because the aggregation can work even if there aren't secondary centers. But if one wants to, if you want to follow this kind of model through, it winds up being something that's reasonably sort of a classic. It's a classic result for this kind of excitable traveling wave model. I'm just trying to see if we're going to get anything that breaks up. Again, because the collisions then let's just let it run for another minute or two. Again, I think you have to decide whether you're going to focus on that the discussion of that is in Stan's thesis, that first chapter on continuous oscillations talks about this issue of spontaneous centers. There was a very famous mathematical biologist by the name of Leon Glass, who published a book on spiral waves and spiral wave chaos. Not so much in the context of dictyostelium. He was interested in the heart and fibrillation, because this kind of excitability occurs also in the heart. But he wrote some very classic papers on the subject. Kaneko in Japan also wrote a lot about this subject, and there's a bit of it in the famous Murray mathematical biology textbook in the second volume. But I thank you for demoing that, because it's really quite a beautiful simulation, and it's pretty dramatic how it behaves. And again, I'm happy to meet with you if you'd like to talk some more in detail about it.
Yeah, I'll keep you updated. Okay, so other updates, I guess. Initialization, I think, is going fine. The write-up is going fine. I did actually have a question about the implementation of the apps. So, like, when we write up things like the biological background, the model explanation, and any exercises, would that go on like the main page of the app instead of like in the app like we do with the Jupyter notebooks?
Yeah, that's a problem with the CompuCell deployment, which is that we don't. The nice thing about Jupyter notebooks basically is the text is live, so you can embed the simulations in the text. For CompuCell, you either have to have an auxiliary document like a PDF file, which has the exercises and the explanations, or you have to put it in the main page. And I don't have a perfect solution. The slide, having the additional materials like a slide deck is helpful. But that's one of the reasons I really did want people to do a little YouTube video to demo it this time, because it's not self-documenting the way Jupyter notebooks are. I mean, not that you can't write a Jupyter notebook that's not self-documenting, but I sort of wish CompuCell had something a little bit like the Star Wars intro where you could scroll text over the front. But we've talked about it. The principal one could pop up a text window. But that's not something we've really played around with.
All right.
So, but that's a good point. And if people have ideas and suggestions about better ways to do that, that would be much appreciated.
And then is there going to be a space on Canvas to submit our project update for this week? I didn't notice one.
Oh, sorry, Josh, could I ask you to set that up? That was my mistake.
Yeah, absolutely. I'll do it. I'll do it right now.
Okay.
Thank you. Okay, thank you for catching that. Okay, let's see, Devin. Drew, why don't we jump down to Drew?
I'm actually on my phone right now, so I can't really show any of the stuff that I have, but I would like to set up a meeting sometime later this week. I think I was sending messages on Canvas, so I don't think they're going through, but I'll send you an email or something to get a meeting going. But
okay, I apologize, I didn't get it. Usually Canvas should forward email messages to me, and I didn't get anything. My apologies.
Yeah, I don't check my Canvas emails that often either. So
I'll just send it through email. But either way, I think my main thing this week was looking at getting ready for my NanoHub delivery and sort of just getting more plots down from the actual paper. But I've got a few questions that I'm going to have about some of the numbers in the paper. I'm not sure how to include them exactly into my model, but I'll have to give you the specifics then.
Okay, that's fine. We'll definitely. I apologize if we missed a message from you.
That's fine.
I'd be happy to set up a meeting. Okay, Fanbo.
Yes, I'll share my screen. Okay, so basically, in the last week, I'm trying to debug my code with the new version of Mechanica because the Mechanica is updated within the clipping plans, and I found some bugs in it and try to solve that. So, and then I have contact with Andy and trying to submit a pull request in GitHub. So, the new version of the Mechanica has already merged my changes, so it can adopt like my Jupyter notebook code for that. And I'm still continue writing documentation function and trying to include a new, like the new packaging one, like with the wrapping with the internal mechanical code. And also in the last week, I'm also trying to figure out how the clipping plans works, and I'm trying to describe like very shortly here. So basically, in the m.int function, you can add a clipping plans argument as that, and basically it would take like several like arrays in it. And for each array, it defines one of the clipping plans. So here, it has two different arrays, so it has two different clipping plans. And you can also like add more clipping plans here. And the first argument inside like one tuple is like a 3D point location, which defines which point should be pointed to. And the second argument is the normal vector of that. So here is a very short demo for that. So here, -1 means a 3D point. So it should be outside of the box location. So it's kind of here. And 1 is the normal vector. So it points out like to the right top of the box. So basically what the clipping plan does is it will cut all the objects that below this line. So everything below this line will be cut. So basically, this function will do nothing to the original simulation box. And I can also show you a very quick demo for that. So basically here, I have only one clipping plans of that. So as shown in the slide, it will do nothing for that. But if I want to change this clipping plans to cut the y direction and only cut the top left of the box, I would just first set a point inside the middle. Because the box dimension is 10, so the middle is 5, 5, 5. And a normal vector points to the y direction. So it will cut everything that points out the negative y direction. Go to this. Okay, so for the next week, the next week task is trying to finalize in the documentation and try to prepare the presentation for next week. And Andy is still waiting for the NanoHub administrator to adopt a new version of the Mechanica, because we have sent the request, but they do not provide any feedback. We don't know about that. And the last work is trying to apply the clipping plans widgets, but since it's not mandatory, and we can and Andy can do it in the summer if we want to apply a grade function set. So I think it's all the function, all the tasks I'm doing this week and next week.
I don't know what the name of it is for the classic model, but I mean, biologists like earth scientists like a model where you take out 1/8 of the image, like cutting a watermelon.
Yeah.
Mm-hmm.
That would be a convenient one to have. I think for example, for for Jeffrey's work, the single clipping plate already would help. But if you actually can bound the clipping plate so you don't go, your clipping plates go from -infinity to infinity, but to be able to define a region so you could do things like cut out 1/8 of a sphere and visualize inside of it the surfaces, that would be a next beyond what you're doing here, but might be a useful one to have.
Yeah, that's actually certainly doable. It would.
What is the name of that display? Because it's a standard. It's a standard one.
I know exactly what you're talking about. I mean, implementing that is actually pretty simple. I mean, basically in the vertex shader, I need to do a little bit of math in the vertex shader itself, and that basically will tell the pixel shader whether to discard that fragment or not. So it's just a matter of basically doing a little bit more geometry. That's actually not terribly big of a deal.
And just looking at Jeffrey's movie images, it struck me, especially for biological simulations, when you show a tumor.
Yeah.
Classic. A cut through the middle is fine, but it's also common to do a quarter, that 1/8 out, so you can see both the surface and the center at the same time.
Yeah, I mean the couple rendering things I want to do after I get this paper out is basically cap off the slices where it's cut, and it's that's actually, believe it or not, it's a much harder problem than it seems like. To basically, it's a matter of generating that texture on the fly to cap it. And I mean, I essentially know how to do it in most cases, but I want to get the paper out first. Yeah,
that's okay.
Yeah.
No worries. I mean, it's just, I mean, I didn't mean to. I
Oh.
understand that these things are not trivial.
Mhm,
I just looking at what Fadmo was showing,
mhm.
that it struck me that might be a next step possibility.
Yeah, I mean, doing a plain octant slice, that's not that big of a deal.
Okay, thank you. I guess that it makes sense after Fadmo to have Jeffrey talk.
I don't have slides today because I had some issues with my computer, but what I've been doing is the chip in the new computer is the M1 instead of Intel, so there's some issues getting some things like Mechanica to run on it. I've gotten it up and going on my Windows desktop in the lab, so I still have access to Mechanica and I have all my files. But I've been talking to Andy about what to do to use it on the Mac, and I think I may have figured out I may have downloaded the wrong version of Python, and there is a Universal 2 installation I just saw, and I think that might be because right now it's running Python on the Intel through the terminal, so I think maybe it that might work. Otherwise, I will just use the Windows desktop version. And I've also started outlining and doing stuff for the paper. So yeah, I don't have much else to say, I guess.
Is this going to be a problem? I mean, not specifically for you, but in general. I remember many years ago when AMD came out with supposedly a much better chip than the Wintel chips. And I bought a laptop which had that AMD processor. And it was true. When it worked, it was extremely fast. Unfortunately, almost nothing ran on it. It was Windows, but even Word didn't work properly on it. Because it wasn't worth anybody's time to develop packages that ran on that particular chip. That was one of my shortest lived laptops because of that.
Well, I mean, I have a universal binary build where basically what you do is when you compile it, you tell it to do a universal, and basically it puts x86, 64, and ARM binary code in the same object file. So basically, it's what's called a fat binary, where it puts both of them in there. Yeah, I mean, it's going to take me a little bit of time to get all of the hardware optimizations for the ARM. Right now, I just have really dumb serial versions of code for that. But yeah,
no worries. As long as Jeffrey can get what he needs done.
Yeah,
that's the only thing that matters from our point of view.
Yeah, I think with Jeffrey, I think he's using the Intel version of the Python runtime, but basically, I mean, our fallbacks are his Windows desktop or actually properly running it on NanoHub proper.
Right. Well, that would be good. If you could ever get them to release it for you, that would be great.
Well, I did put a fire under Nick's whatever today, and evidently he is moving forward on getting a image on production.
Okay. Well, we don't. Thank you. That's fine. So I guess Tyler, I think you're the last, right?
Yeah,
Did I miss somebody? No? Okay, go ahead.
I don't have anything new this week to show, but basically my model is as close to it can be as it can be according to the information given in the paper. And this week I'm going to focus on debugging it. Most specifically, with my model, the behavior of cast space in my model is not as expected in CompuCell. So I'm going to work on deconstructing it and simplifying it in order to get to the bottom of that, and then hopefully have that working in a few days. And then hopefully by this week, I can get the NanoHub, or start working on getting it, the project uploaded on NanoHub. But that's pretty much all I have that's new, just debugging.
I mean, for next week, if you have to upload it, in other words, run the base CompuCell v4 and then upload it and run it that way, that's okay for next week. In other words, I want to see it running on NanoHub, but it doesn't have to be loaded as an app on NanoHub next week if you can't get to that.
Okay.
Try, but if it turns into a misery, don't focus on that. Okay. And if you want to meet more to try to do some more debugging, I'll do my best to be available.
Sure.
Okay, I think that's everybody. I'll turn it back to you, Josh.
Okay. 1 second. So James just mentioned it, but just as a reminder, the first version of the projects are due next week. As a starting point, having those of you who are working on CompuCell, as James mentioned, having a working version that you can upload into the NanoHub CompuCell tool and run it, I think would suffice, because we haven't showed how to upload the CompuCell simulations as tools. That might be something we might want to organize with Giuliano next week, so we can showcase how to do that. So James, if you want to email Giuliano, that would be useful. Okay, so last week we've been talking about how to integrate different scales of biological processes and objects that span over different, and how to integrate models that count for objects that span over different scales. Some of those processes and objects might pertain to intracellular things that are going inside the cell that control the cell behavior and the cell specific phenotype. Some of these processes might be happening at the organism level, where or for example, compounds might be transported across the body and be passed on or been an input into models at a tissue level, or processes that occur at a tissue level. And so far we've focused on those processes that pertain as intracellular signal or subcellular signaling. One of the examples that we have discussed extensively, and that we will come back to it at the end of class today, is the delta notch pattern. I'm not going to go into the details now, but we will come back to it. Another example is a cell cycle model. And we've mentioned that so far the way we have modeled cell cycle, or cells undergoing proliferation, is by having their target volume increase and then triggering a cell division event when the cell reaches a specific volume. But we can be more explicit about how we model the cell cycle by modeling some of the molecules that regulate the cycling through the different stages of the cell cycle. And that's in that specific case, the cell division and proliferation would be a emergent result or an emergent property of this explicit model of the molecules that regulate the cell cycle. And if we have time today, we will come back again to we'll go over the delta notch, but we will try to get to a more explicit model of cell cycling. We presented a workflow of how to integrate these intracellular models into the cells, into CompuCell cells. And this is important because these tips relate to what James was talking about earlier, the need of implementing the models piecewise, in the sense that you want to have the intracellular model as a standalone that you can explore. You want to be able to run it, preferably using anti-money model, but any SPML solver will do. You want to understand and have an idea of how the parameters of the model interact between each other, what the dynamics of the model are, and what are the potential parameters that can be connected to the spatial counterpart, right? Once you put it in the spatial context and the multicellular context, what are the parameters that would allow you to link the intracellular model to the spatial information or the cellular information? But to be able to do that, you need to have a good idea of what the dynamics of the model as a standalone is or are, and what are the parameters that you might want to control or might want to link to other properties in the cellular pods model. Then there are, once you have your SPML model, you want to load it into the cells of your simulation. And this is another step that James was talking about earlier, but this idea is important. We were talking about it earlier, that you might want to do so again, step by step, by first initializing it in a single cell. And as you initialize it in a single cell, then you have a control over the parameter that you will be linking in the future to the a output or of your CompuCell simulation. Either it be, for example, the number of cell neighbors that the cell has, or the chemical field that the cell is exposed to. And then you want to be able to control the inputs of the intracellular model without linking that model yet to the cellular, to the any other cellular component. And once you have that model running and you know what it's doing, then you can calibrate, then you can start linking your model to the other aspects of the CompuCell simulation by making sure that the parameter that you were inputting, that is being inputted into the model, is being properly scaled and calibrated, so the proper dynamics of the intracellular model so it results in the proper intracellular dynamics. Then we can, once you've calibrated that intracellular model, then you can time step it, and then you can rewrite and read and write values by using a simple SPML syntax that we will come back to it later. James talked a little bit about how to specify models in antimony. I'm not gonna go back into the full details. I'm gonna put in the link. I'm gonna put in the chat a link to the tellurium documentation later. It's very useful. But the idea is that the quick idea is that we basically define reactions as something arrows something, S goes to R. That's how we call it, and it's that indicates that the species S is being transformed into R at a rate that is specified after that is controlled by parameter K1. And then so on with the other reactions corresponding to the model. And then we have to specify what the values of the parameter are and what the initial conditions are. And those are the basic elements of the antimony model. More complicated, those who took class with us last semester know that already that there are more complicated elements that we could include here, but this is just the basic, and this is just the basic of the antimony model specification. Once we have the antimony model, we can load it. There's different options as to how to load the antimony model. You can add it to at a specific cell. You can add it at a specific cell by its ID, or you can add it to all the cells that belong to a specific cell type. Or you could also have it as a free-floating model. And the idea of a free-floating model is one that we'll probably come back to it later, but it potentially has two applications. You can use it as a way of linking whatever is happening in the simulation to processes that are happening at scales higher than the tissue level that you're simulating with CompuCell, or you can also use it, and we often do, as a way of calibrating what the model does by itself. Let's say the intracellular model does by itself versus what it's doing inside one cell when you put it and you instantiate it inside a specific cell. There is a very simple format and syntax as to how to time step the model. Once we have done everything that we want to do with our models, we just call this time step _ SBML method, and that will just make sure that the models are time stepped by the time step that we've set up in our when we've loaded the model. And then we can both write and read the values of the SBML model just by referencing using the SBML.model name syntax. In the case of a model that is inside a specific cell, we do so by the prefix cell. If it's a floating model, because it belongs to the whole simulation, we use the prefix self. But the idea is that we then call the model by the name, and then we specify inside of parentheses, inside of bracket parentheses, what the name of the variable inside that model is that we want to retrieve or that we want to write. And then this is a function that we sometimes use, and we have it in our viral infection model. You also have the option of copying the SBML model from one cell to another, and you have an option of deleting the SBML model from a cell. Let's say your cell died, and that means that it no longer has an SBML model inside of it that you care about. The probably safest way of unloading that model as well as to make sure that it's not doing anything that you don't want it to do is by deleting that SBML from the cell. Okay, We'll probably come back to it. But now I'm just gonna pass it on to TJ, who's gonna be talking about how to implement the Python API for CompuCell. Sorry.
All right. Well, hello, everyone. Can you hear me okay? Great. Okay. And can you see my screen? Great. Okay. Yeah, I'm very happy to be here today. This is a topic that's near and dear to my heart. So I'm always very enthusiastic to get to talk about it. So today in this portion of the lecture, we're going to talk about how to leverage the CompuCell3D Python API to do execution of simulations within a generic Python script, and how to get information from a Python script into a CompuCell simulation and to get information back. These operations should be mostly familiar to all of us by now, if we consider some of the basic operations that we've been doing in some of our CC3D model scripts. So, you know, we write our models in XML and then in Python for our steppables. And oftentimes we find in our steppables that we would like to use other Python packages like the random package, in which case we go to the top of our script and we type from random and then import something, import rand int or import random. So this is a common sort of run-of-the-day operation to import functions and classes from other packages and use those in our own scripts. Today, we would like to accomplish learning the capability of doing that with CompuCell. So start with a generic Python script and say from CC3D and import something. And then with the thing that we import, execute our simulation and interact with our simulation, do things with it. And we will do that through the CC3D API. And you can compare this to how we're used to using CompuCell so far. So ordinarily, we'll open up Twedit. We'll use the wizard to generate a new project template. It'll create our XML. It'll create our steppable script, you know, and we sort of fill things out from there and does lots of, you know, the sort of behind the scenes stuff that we don't like to have to worry about. Now with the CC3D API, instead of relying on, say, player to load our simulation and then to execute it interactively, which is nice and has lots of benefits. Instead, what we can do is we can take our generated project from Twedit. We can develop our model in Twedit. We can instead edit it in some other editor that we might like PyCharm, for people who do lots of Python development. And then instead of going over to player and loading our simulation, instead, we'll write a Python script that imports the necessary classes from CompuCell. And with those classes, then instead we'll execute our simulation from our Python script. So the workflow is a little bit different. There's lots of compatibility between what's generated in Twedit and what can be used with the CC3D API. The data that's generated is all the same. So anything that's been executed through the CC3D API can then be, say, loaded into player and reviewed thereafter. We're just going to do things a little bit different today, and more on the programming side. So I would like to sort of give you a sense of what the purpose of this functionality is in the first place. And that's sort of back to sort of the basics of the sorts of, I would say, elementary things that we construct in the source code that we write. You know, you can imagine constructing some generic function. I have one listed here as just a Python function called myFunction. And myFunction just takes some argument of x. And you can imagine it does something here with x. And based on what's given to the function through x and the calculations, our function called my function then returns some variable y. And y is a result of the calculations that are done that involve the information that's given to the function through x. Now, what we can do with the CC3D API is we can essentially do this exact same thing, but with our CompuCell simulations. So we can do things like construct functions that can take information from, say, something else that you may be developing. You may be working with, say, an optimizer or with artificial intelligence or image segmentation. And you can take information from something else in your software, and you can give that information as an input to a simulation that you have already developed in CompuCell. And then your CompuCell simulation does the things that we're all well familiar with now, the things that occur when we execute our simulation. And then at the end of that simulation, the simulation returns information back. It gives some sort of output, some set of data that's a result of the inputs to our CompuCell simulation and the execution of the simulation itself. So we can do things like write functions that will take arguments, execute a CompuCell simulation, and then return information back. And that enables, like this simple little diagram here, all sorts of workflows and functionality to be able to take a CompuCell simulation and essentially inject it as one component in some broader workflow, like doing model calibration using an optimizer and CompuCell3D. Now, not much changes when it comes to how all of these projects are set up. So we ordinarily we'll open Twedit, we'll use the project wizard to create our sort of template. And this template, when we look at the actual file structure, can be described for this example of a project called My CC3D Model. If you run through the project wizard and tell them that your simulation name is My CC3D Model, then it will create this corresponding file structure. There'll be a CC3D file, a simulation directory will be created, and then we have the scripts that we're familiar with. The XML describes the CC3D ML for our simulation. The steppable script has our steppables, and then the main Python script brings it all together and passes our steppables to CC3D. Now, to use CompuCell simulations in using the Python API requires absolutely nothing different from this file structure. So what is generated by Twedit can then be used both in player and externally in another Python script through the Python API. And so to create a project like this that uses a simulation that's been constructed externally in some other Python script is simply a matter of just creating some directory that contains everything that's generated in Twedit. And then in the most simple case, just an additional Python script. And that Python script will do all of the importing of the CC3D API, giving our simulation to the API, performing execution, and whatever other manipulations we'd like to do. So there's nothing extraordinary that has to be done when taking something that you would already generate in Twedit. Instead, it's just adding additional things to do with that simulation. So setting up a Python script to be able to execute CompuCell3D through the API does require a couple of prerequisites that are ordinarily done for you by player. And this is a very short list. This may look like a lot of information, but overwhelmingly so. The bulk of the information is contained here. So there are a couple of environment variables that have to be set for CompuCell to know where all to find things. So for those of you who are following along and would like to actually go through the exercise with me, you may want to make a note of these. We'll refer to them again, but Most importantly, there's an environment variable called prefix underscore CC3D. And this just has to be set to the path to the directory that contains your CompuCell installation. Secondly, there's an environment variable called Python install path, Python underscore install underscore path. And this is the path to the directory of the Python executable that's distributed with CompuCell. So this is actually within the installation directory. So this is no big deal either. For Linux and Mac users, there's an additional one that's optional, but does cut down on some of the clutter that's printed to the console, and that is this environment variable called lc underscore numeric, and that's just set to this string here, c.utf-8. Windows users don't have to worry about this. Mac and Linux users can ignore it and just ignore some of the clutter that may get shown towards the beginning of a simulation. And then lastly, there is a path that has to be appended to the Python path. We'll show how to do this in Python in one of our Python execution scripts. But the path that has to be appended to the Python path is whatever is given to prefix CC3D in the environment variable, and then adding on to the end the subdirectory lib/site packages. And that is it. So this can be set up in some sort of execution script. Windows, Linux, and Mac, they all have their own scripts that you can write to execute from command line. This can all also be accomplished in a Python script, which is what we'll do today. But this is sort of one of those things where you kind of make a note of it, you make a template script and store it somewhere on your computer, and then every time you create a new simulation project, you just sort of copy this in over from some template. And for your reference, if you have a hard time tracking this information down, you can also just go to your CompuCell installation directory. I'll show you an example from my computer. I'm on Windows, and I'm looking here at my root directory. So this would be the directory here that we're looking at would be what I would pass to prefix CC3D. Now, you see we have these scripts here for CC3D, CompuCell3D, so on and so forth. If you actually open this and look at it, you'll find that these variables have already been set in your installation. So you can actually also, instead of finding this stuff manually, you can just go to one of these scripts in your installation and just copy that information. Okay. Give everyone a second. We have a poll going. Make sure, check in with everyone, make sure everyone's following along before we proceed. Again, this is something we'll revisit, and by and large, after having accomplished this once, you never really have to worry about it all that much ever again. All right, we've got 2 responses and 3 responses. 3 of 3 say they're good, so let's proceed on to the more fun stuff. So executing a CompuCell simulation from Python all occurs through one class that's provided in your CompuCell simulation or installation, and that class is called CC3D caller. So if you are writing a Python script to execute CompuCell from within that script, what you would look to do is to instantiate a class instance of this class CC3D caller, and you'll give all the information to CC3D caller to execute your simulation according to whatever specifications you'd like, and all of the operations to execute the simulation and get information back will all occur through that instance. And you can see an example of what this looks like in the code snippet here in the bottom. So CC3D caller can be found in your installation at cc3d.compuscellsetup.cc3dcaller. So that is a class that's defined within a Python script called cc3dcaller.py. So to instantiate one of these looks like this code snippet here. So we're importing cc3d caller, and then we're going to instantiate an instance of cc3d caller, giving it some information, and then interact with our simulation through that cc3d caller instance. Now, the most basic inputs to execute a simulation through the Python API, there are three. The first is has the keyword argument cc3d_sim_fname, and this is the absolute path to the cc3d file of your simulation script. So this is a script that ordinarily is generated by Twedit. This is what you load when you load a simulation into Player or into Twedit. You select the cc3d file. We're going to pass the path to that file to this cc3d caller class with the keyword argument of cc3d_sim_fname. Now, the second basic input is an integer that specifies the frequency of output. And this is an optional argument. So if you don't actually want to output any data from your simulation, then you just wouldn't pass this. But it's pretty typical to do this. So I included it here in our basic overview. So we can specify a frequency of generating, say, lattice data from Player through preferences through the Python API. We do this by specifying an integer value and passing it to the keyword output_frequency. And then lastly is a path to a directory for where to save the data that's generated by our simulation. So when we specify an output frequency and CompuCell goes to start outputting information at that frequency, it will do so at the absolute path that you pass to this third argument, which is output_dir. So we pass three things: the path to our simulation file, we pass a frequency at which we'd like for CompuCell to generate data, and then we pass an absolute path to where we would like to store all of the generated data. Now, after having accomplished this, this instantiates an instance of cc3d caller, and all that's left to do to execute the simulation is just call the method run on that instance. Instantiate it, and then just call, in this case, cc3d_caller.run with no arguments. And if you watch your console from there, you'll see the bells and whistles go off, everything lights up, and you start seeing it spitting out data, outporting debug information at regular intervals, so on and so forth. And then as soon as the simulation finishes executing, then control is returned back to your Python script, and then your Python script continues execution after that call. And that's it. That is the most basic example of using the Python API to execute a simulation from within Python. And so let's stop here and let's kind of do a crash course through how to do this. So we're going to start from an empty directory. We're going to grab a demo that's included in the cc3d installation, and we're going to set up all of our scripts, and we're going to do this very basic application with the API to just execute our simulation and confirm that it works as intended. So I am going to put our work list here on the left. These are all of the steps to do this. I'm going to do this here on the right. I'm working in PyCharm. You're welcome to do this in Twedit. Either way, just follow along. If you have any issues, of course, please just speak up or put something into the chat. We'll try to push everything along. If we run into major issues, then of course, everyone has my email, and I'll include it again. We can work offline after lecture. So to start, what we're going to do is we're just going to create a directory somewhere on our drive where we're going to do all of our work for this exercise. And I've already created mine. So I have this directory that I've created. It's empty. And I'm just calling it CC3D 2D cell sorting. You can name it Pancake if you want. It makes no difference. We're just going to have a directory to work in. And this is where we're going to do everything, is in this one directory. Now, within this directory, we're going to create another directory that's going to hold some of the simulation files that we're going to grab from the CompuCell3D installation. We're going to name this folder, this does matter, we're going to name it cell sort, one word, no uppercase, and then underscore 2D, the D being uppercase. So if you're following along, it's cell sort underscore 2D. We're creating that in our project directory for this exercise. And you can just leave that empty for now. Now what we're going to do is we're going to go into the demos that are provided in CompuCell. So if you go to your main CompuCell installation directory and then go to demos, the fourth directory down should be Callable CC3D. Let's go in there. And you'll notice that there's another folder created in here with the same name as the one we just created, cell sort underscore 2D. So from the main installation directory, you're going to demos and then Callable CC3D. And you should find a folder called cell sort underscore 2D, just as we named in our project directory. So we're going to go in there and we're going to grab 2 of the 3 items that we should find there. We're going to copy the simulation directory and the CC3D file. So in that directory, you should find a folder called simulation and a CC3D file called cell sort underscore 2D. This may look familiar. This is a project that was generated with Twedit. And we're going to copy that, copy both of those. And we're going to paste those in our cell sort underscore 2D directory in our project. So last review, main installation directory, demos, callable CC3D, cell sort underscore 2D. Grab the simulation folder and the CC3D file named cell sort underscore 2D, copy both of those and paste them in the cell sort underscore 2D folder of our project folder for this exercise. All right, now let's go back to our checklist. That is steps 1 through 3 accomplished. The next thing we're going to do is in our main project directory for this exercise, we're going to create one more directory. We're going to name it results, all lowercase. So in our main project directory for this exercise, we're going to create one more empty directory. We're going to name it results, all lowercase. This results directory is going to be the output directory that we pass to our CC3D caller instance. Okay, so if everyone's with me, next thing we're going to do is we're going to create our Python script that's going to do all of the execution of our CC3D simulation, but outside of player. So we're going to create a Python script in our main simulator, or our main project directory. So if you named your folder cell sort 2 or CC3D 2D cell sorting like I did, and that's the folder you're going to target, you're going to create a Python file in that directory. And I'm just going to name mine CC3D underscore 2D underscore cell sorting. The name mostly doesn't matter. We're only going to very briefly reference the name of this file towards the end of this exercise. But by and large, the name of the file makes no difference. It just needs to be in the main project directory for this exercise. Now this script needs to accomplish 3 things. And we can put some comments to start our script to sort of demarcate what work we will do where. First thing it needs to do, we can put this in as a comment, set up environment. And we'll do that somewhere after that comment. After having accomplished that, we're going to set up CC3D simulation. We'll do that somewhere after that comment. And then our third step will be execute CC3D simulation. Those are the 3 things to accomplish with this Python script. Now, to set up in the environment, we have to accomplish the boring things that were described earlier on in the slides. And we can do these as follows. So to set environment variables, we can use OS. So let's start by, at the very beginning of our script, import OS. And while we're at it, since we'll be appending something to the Python path, we'll also need sys, import sys, S-Y-S. So import OS and import sys. So the 2 modules we'll need to set up our environment. Now to set environment variables from a Python script, can do something like this. We'll be using OS. It'll be os.environ, E-N-V-I-R-O-N, os.environ. Now, os.environ can interact with this like a Python dictionary, where the keys are the names of the environment variables, and the values are the values that correspond to those environment variables. So for example, the very first environment variable that we discussed earlier on was the environment variable prefix underscore CC3D, all caps. We can set the value for that environment variable using os.environ, where prefix CC3D is the key. And if you recall, prefix CC3D needs to be set to the path of your CompuCell installation. So in my case, it looks like the following. I just have mine installed in my C drive, and it's C:/CompuCell3D.py3.64-bit. You may have to do a little rummaging in your file explorer of choice to find what that path is, but you'll want to set that to something like what is shown on my screen in my file.
TJ, if you allow me to add something up here, usually for people who installed on Macs, you'll find the installation in the main application folder, which is usually which usually, as I typed on the chat, that's usually the prefix to that installation folder. So for those in Macs, it will of course look a little bit different. And I'm just, yeah, the default. It might be installed in a different folder, but it's usually installed in the application folder.
Thank you, Josh. Okay, so then the second environment variable to set, we'll go ahead and start an os.environ. We're going to interact with it again like a dictionary. This time, the second environment variable to set, again, all caps, Python_install_path. And Python install path is the directory that contains the Python executable from within the CompuCell3D installation in general. On Windows, if you go back to the main directory, and this will be true on Mac and Linux as well. For Windows, currently we use Python 3.6, and the directory will be named Python 36. For Mac and Linux, it's Python 37. But Python install path just needs to be the path to this directory. See, there's my Python executable. This is the Python executable that's used to execute CompuCell simulations. So we just need to add that path, assign that to the environment variable Python install path. So for me, it looks like this. My prefix CC3D value, and then one more subdirectory, Python 36. And then the last thing to accomplish for setting up any CompuCell environment in Python is adding something to the Python path. In general, we can do this with sys. We can do this by saying sys.path.append. Now, whatever path we pass to sys.path.append, this is appended to the Python path. And if you recall, the path was the value that's passed the prefix CC3D, and then two subdirectories down from there was lib and then site packages. So it'll be the same thing as what you assign to prefix underscore CC3D, and then two directories down, lib, and then site dash packages. And that's it. From here, you can import anything from the Python CC3D module and blow things up from there in your simulations. So, so from here, now the next thing to do is actually get our CC3D caller class. So we're moved on to our second step, which is to set up our CC3D simulation. And if you recall, the way to do that was to start from and then CC3D, all lowercase, and then dot CompuCell setup. You may want to check my screen for upper and lowercase. The C's in CompuCell are both uppercase, and then setup, S in setup as well. So from cc3d.compuscell setup dot cc3d caller. So this gets us to cc3d caller.py, and we can say import cc3d caller. That is how we get CC3D caller from CompuCell. Okay, so then now let's do a little setup to actually give CC3D caller our CC3D file path, as well as our output directory, which is the directory we created called results. So let's do a little bit of declarations here. Let's first just keep it simple, and let's just declare a variable that refers to the path of our main project directory for this exercise. Let's call that variable project_dir. Project_dir is going to be the absolute path to our main directory. We can do this by saying that that is equal to, follow with me here, it's os.path.dirname, dirname, and the variable we're going to pass to it is something built in, and it's 2 underscores, file, and 2 more underscores. This command here, os.path.dirname, and then passing __file__, this is the absolute path to the directory that contains the script that we're working in. And in our case, since we declared this script within our main project directory, project dir will be the absolute path to our main project directory. And now from here, let's declare a variable that is the absolute path to our CC3D file. So we can do this. Let's declare a variable that's the same, has the same name as the keyword argument that we're interested in, and that is CC3D_sim_fname. And that will be equal to, follow with me here, os.path.join. I'm going to create a path by joining a couple of absolute and relative paths. So this will create a consistent absolute path based on the arguments that we give it. We're going to start by constructing our path from our variable project_dir. So this will be the start of the path that we're constructing. And then if you recall, our actual CC3D file is in the directory cellsort_2d. So the second argument will be the name of that directory as a string. So this is going to take our project directory. It's going to add cellsort_2d subdirectory to this path. And then the next thing we'd like to add onto this path is the name of our CC3D file. So our third argument will be another string, and the name of that CC3D file is cellsort_2d.cc3d. So the CC3D_sim_fname, this will now be the absolute path to our CC3D file, and that's constructed by appending to our project directory path the subdirectory cellsort_2d, and then the file name cellsort_2d.cc3d. And then lastly, let's also construct an absolute path to our output directory. Our output directory will be the results directory that we created in our main simulation, our main project directory, the directory called results. So let's declare a variable here with the same keyword name that we're targeting, and that is output_dir. And you may have guessed it, we're going to do os.path.join, and we're just going to construct this from the project dir variable that we declared, the absolute path to our main directory, and then we're going to append to that the name results, the directory we're interested in for outputting data. And so we're going to do that by passing a string as the second argument to os.path.join, and the name of that string, or the string is the name of the directory results, all underscore. So now we have most of our inputs constructed. Absolute path to our CC3D file, absolute path to our output directory. This is 2 of our 3 basic inputs. This is sufficient to go ahead and create our simulation instance. So let's do that. Let's do next line or two. I'm going to be on line 17, CC3D underscore caller. Start a variable named CC3D underscore caller. And we're going to assign that to be equal to an instance of our CC3D caller class that we imported at around line 11, in my case, CC3D caller. And let's start some arguments to instantiate this class. Now again, the first keyword argument is the absolute path to our CC3D file. Out of convenience, we named it the same as we named the variable that contains that value, CC3D underscore sim underscore fname. That has the same name as the actual keyword. So we're going to do something that maybe look a little silly, which is CC3D sim fname equals CC3D sim fname. First one being the keyword, the second one being our variable. There's no convention that's being adopted here. The name of our variable could have been potato. We're just going to keep things nice and simple. Now, the second basic input, or second argument, is the output frequency for our simulation. And that was output underscore frequency. That's the second keyword argument. So we'll do output underscore frequency equals, and we're going to pass an integer for the frequency. And let's say we're going to output at a frequency of 10. So every 10 simulation steps, our executed CC3D simulation is going to spit out data and dump it into the directory that we specify in our last basic input argument, which is the output directory, the keyword being output underscore dir. Again, no convention here, just happen to name our variable that contains our output directory path, just happen to name it the same as the keyword argument. So here we're going to say output underscore dir equals output underscore dir. Now, if you're here with me, keeping up, this instantiates our class. This is all of the setup required to do this very basic execution of a CC3D simulation in Python. The very last thing to do is to actually do the execution. So in our last section for execution, I'm going to do our method run on CC3D underscore caller. So our instance of CC3D caller, we're going to take that variable and we're going to call its method run and pass no arguments. And that's it. Now, to actually execute this, there are a number of ways that you can go about executing a Python script. We're going to do this in a way that we typically do when building these sorts of projects, and that is that we're going to create a very simple execution script in our project directory and we're going to have that execution script just execute this Python script that we just wrote, and we're going to execute it using the Python executable in the CC3D installation. So in your main project directory, you're going to create one more file. Again, you can name it whatever you want. I'm going to maintain a little bit of consistency here, and I'm going to give it the same name as my Python script, CC3D_2D_ cell sorting. Now I'm on Windows, so the file that I'm going to create has the extension BAT. If you're on Mac, your extension will be .command. And if you're on Linux, then you can create a shell script with the extension .sh. So we're going to create this execution script. Just have a blank file here. And what we need to do now is to call the Python executable in the CC3D installation and pass to it the name of our Python script. Now this looks a little bit different per operating system, but there are equivalents on Linux and Mac to what I'm about to do here. So if you do run into trouble, this is a fairly quick fix. I'm just going to demo it on Windows. I'm going to grab the path to where our Python executable is, and this was the path that we gave to the environment variable Python install path back in our Python script. I'm just going to grab that path because that's the directory where the installation is. And in a batch script, I'm going to start some double quotes, and I'm going to paste in that directory as such. Again, this is the path to the directory that contains the Python executable, which means that then I can get the Python executable by typing /python. On Windows, it's python.exe. So this will call the Python executable in the CC3D installation. I'm going to start a second string that will be the argument that's passed to Python, and the argument will be the name of the Python script that would like to execute. So whatever you named your main Python script where you wrote all of your execution specification, I'm going to pass that as a second argument, and we're going to do that in double quotes as well. In my case, it was CC3D_2D_ cell sorting .py. On Windows, if you want to cut down on some of the junk that will be output, you can just do ampersand echo off beforehand. Not important, just keeping things a little tidy. And that is it. We can now open up a command prompt. We can execute this script from our command line. And I'm just going to do this now and see what happens. So I'm going to go back to my project directory. Your project directory should look something like mine. We've got cellsort_2D that contains the simulation that we grabbed from the demos. Results is the output directory that we created. That's where our output is going to go during execution. Python script that actually imports the CC3D API and uses it, and then a simple execution script to actually do this. So I'm going to open a command window in this directory. So the working directory to execute this should be our main project directory on Windows. Hold Shift and right click and then open command window here. And then CC3D_2D_ cell sorting. Voila. So some of this output might look familiar. Maybe I've noticed that this is the sort of stuff that's printed to the console in player. In this case, actually seeing that information printed here in the command prompt. We've got a little message here saying the calls were finished, and then the same sorts of performance reports that we can find in a terminal when we're running player. Now, if we did everything correctly, then we should find that when we go back to our main project directory and go into the directory results, we should find a folder called lattice data. If we go in lattice data, should find some VTKs at a frequency of 10 simulation steps.
Yeah, TJ, since we haven't explained what the VTKs are beforehand, these VTKs record, well, you might want to explain them quickly if you want.
Sure. Yeah, so what these VTKs are, this is essentially the spatial configuration of your simulation at various points in simulation. So CompuCell knows how to take this information and recreate your simulation at these various time steps. It's basically like a snapshot of all of your spatial data. So each of these VTKs contains the current state of the cell field, whatever PDE fields you may have been simulating, so on and so forth. It's all stored and with the suffix of the simulation step at which these snapshots were created. So this is the same data that's generated if you were to do the same procedures but in player, using preferences by setting an output frequency. This is all exactly the same, which means that we can then go to player and open this up and see what it looked like. So let's, everyone who's following along, let's open player. And I'm going to navigate to, well, let's start over. Let's go to the file menu. And we're not going to open a simulation file. We're going to open a lattice description summary file. This would be the second item in the file menu. So open lattice description summary file. That's going to take us to a file browser. Now we're going to navigate to wherever you happen to have saved your project directory for this exercise. In my case, I actually have them stored somewhere buried in my CompuCell installation. I don't recommend doing that. Okay, so now I've located my main project directory for this exercise. We're going to go into that folder, go into results, our output directory. Now we're going to go into the lattice data folder that was created by CompuCell. You should see that there is a file available with the extension DML. Let's select that and open it. So in the results directory, go into the lattice data directory and select this file stepldf.dml. Select that and open it. And then in player, you can just press run. And here in the left of player, we have this little pane for lattice data files. You should notice that all of these files that are shown are the VTKs that we very briefly discussed a few minutes ago. And then each of these corresponds to a result that we're seeing here in this rendering window. So the same functionality as with running a simulation is available when reviewing results like this. So you can create graphics windows and render data and step through it. Or you can just hit play. The only difference here is that this is replaying simulation data that was generated previously, as opposed to rendering data that's being generated in real time. And that's it. That is how to get from an empty Python script all the way to reviewing results in replay using Player. So for those of you who didn't keep up, these simulation scripts will be available, so you can walk through this after class. And again, please don't hesitate to contact me to walk through some of these steps again if you'd like to go through this on your own and are struggling. With all of that being said, we're going to continue on to the really cool stuff, at least I think, and that is getting information into and out of a CC3D simulation. So the way we can think about a CC3D simulation in a generic Python script is we can think of it as a black box with an interface. So just like the very simple function that we discussed very early on when we first started discussing this topic about a generic function that takes an input as an argument and then does some calculations and then returns some resulting output, we can do the same thing with CC3D simulations using the CC3D API. And we're going to discuss this with some terminology to sort of try to keep these ideas clear. And especially what we would like to demarcate is the computations and specification that we do inside a CC3D simulation versus the things that we do outside of the CC3D simulation in a more generic Python environment. And so we're going to call these a CC3D space and a Python space, where a CC3D space is, say, the space where we have been doing CC3D model specification all the way up until this point in this course. When we create our steppables script, we write our steppables and we register them with CompuCell all within Twedit. That is the CC3D space. And where we just were, where we imported CC3D caller after having set up our environment variables and then calling run on our CC3D caller instance, we'll call that the Python space. So there are two sides here. In the Python space, we are instantiating instances of CC3D simulations. In the CC3D space, we're designing steppables. We are interacting with individual cells, interacting with fields, doing secretion, so on and so forth. Now the interface between these two, passing information from the Python space, as shown here on the left, something like what we just accomplished, creating a CC3D caller instance. Here in the Python space, we would like to take information from this space and pass it into the CC3D space and use that information somehow as part of the calculations that are done in the simulation. And then from the CC3D space, return some result back over to the Python space. In this example here, this is something like choosing a cell ID and then passing that ID to a CC3D simulation through our CC3D caller. Give that ID to the CC3D caller and then get that ID somewhere in one of our steppables, and then get the corresponding cell that has that cell ID that was specified in our generic Python script, and then pass that cell back to our generic Python script, back into the Python space. So that's the information pipeline that we would like to establish here. information to and from these two different spaces. So to get information into CC3D, say from working from the script that we just designed a couple of minutes ago, we can do this with an additional argument that's defined for CC3D caller. And that argument has the keyword sim_input. So when we instantiate a CC3D caller instance, we can also pass something defined in our Python space to the keyword argument sim input, and whatever is passed can be retrieved in the CC3D space. This could be something like a cell ID. In this very simple example, it's just an integer. More importantly, this can be a Python dictionary. This can be, it's maybe dangerous to say anything, but these can be containers that have all sorts of information. So in one object, one Python object, you can bundle just about anything that you want and pass it to sim_input when instantiating a CC3D caller instance. And whatever you pass there in the Python space can be retrieved from within your CC3D simulation as follows in this code snippet. The first thing that we do is we get what's called persistent globals. Some of you may have interacted with this already with some simulations. What we say is that from cc3d.CompuCell_setup, this is in a steppable script, from cc3d.CompuCell_setup, import persistent_globals, and whatever was passed to the CC3D caller instance can be retrieved as the object on persistent globals with the name input_object. So now in our example here, say we have a cell ID that we've selected in some Python script in our Python space. Say we're selecting a cell ID of 1. We'd like to pass this cell ID to our CC3D simulation and then design our simulation steppable such that the steppable can receive or get this cell ID that was passed to CC3D caller and then fetch the corresponding cell by ID. We can get the ID that was passed through the Python API by including at the top of our script from cc3d.CompuCell_setup import persistent_globals, and then get the cell ID as persistent_globals.input_object. Now all of this will be accomplished during execution of the simulation, which is this last line shown here in this code snippet. Now, returning information is a little bit different, but about as complex. Our CC3D caller instance actually returns something when we call run. We've been executing simulations by calling something like cc3d caller.run. And up to this point, we've not really considered that that actually does return something, but in fact it does. And what it returns is a dictionary that has a very special key and value. The key result in this returned dictionary contains information that's passed back to the Python space from the CC3D space. So if we start from our CC3D space and we ask how can we return something that's available back in the Python space, we can also do this using persistent globals. From cc3d.CompuCell_setup import persistent_globals. To make something available in the Python space, we can use a second object on persistent globals, and that is named return_object. Persistent globals.return_object, whatever we set this to will become available in the returned dictionary by CC3D caller.run. And it is specifically accessible with the key result. So when I assign something to persistent globals.return object in my CC3D space, in say one of my stepables, and then I execute this simulation using CC3D caller and get the returned dictionary, I can retrieve what this CC3D simulation returned. I can get that in the dictionary at the key result. So in this example here, to sort of conclude how to fetch a cell from a CC3D simulation, but all from a Python space. Again, we're passing a certain integer cell ID. We're passing this ID to a CC3D simulation with the expectation that it somehow will fetch the corresponding cell at the end of simulation and return that cell back to our Python space. We would set up our CC3D simulation script to do this lastly as follows. This last line here, up to this point, we've retrieved the cell ID that was given to this simulation from the Python space using persistent globals.input object. We then have the elementary operation of fetching a cell by the ID using the Python stepable method fetch cell by ID. This gives us a cell in our stepable, and then we can return that cell and make it available in the Python space by assigning it to persistent_globals.return_object.
TJ, I have a question.
Sure.
In the as a simulation input, you're passing here from the Python space a integer. But what other types of objects could you pass in as a inputs that can be read off from the persistent globals?
Like anything else in Python,
Look, I could pass a dictionary or a list or whatever I want.
Yes, and I mean it's to the extent that you could pass information from a previously executed CC3D simulation. You could pass, say, information from image segmentation to initialize the cell field at the beginning of simulation. You can pass functions. I mean, it's hard to describe what you can't. In general, the strategy that I typically employ is to take whatever information that I would like to give to my simulation as an input and package all of it in one dictionary. And then at the very beginning of my CC3D simulation for my stepables, I'll go through a whole process of actually unpacking that dictionary and processing all of those inputs with however those inputs affect my simulation.
Okay, yeah, thank you.
And the same is true for returning information. And this is demonstrated here in that we're taking a Python object that is intrinsically CompuCell, which is the cell, the cell G instance. These cells have attributes like a target volume, all sorts of SBML models that have been attached to them, things that are very CompuCell specific. But then we're passing one of these back to the Python space. And so in the Python space, we now have a CompuCell object It's still attached to this simulation in many ways. For example, we say things like self.cell list. This cell is still attached to that cell list, but we can still do operations on these different CompuCell objects, but now in the broader Python space. So this is the information pipeline. Pass things in through sim input from our Python space. We get those inputs in the CC3D space using persistent globals.input object. We do whatever we'd like in our CC3D simulation, and then what information we would like to make available back in the Python space, we can assign to persistent globals.return object. And so this leads us to our second exercise, and this is one that I could probably spend hours on. So I thought it more appropriate to share with the class. You can review this at your leisure and at your level of interest. But there is a demo that I would like to bring to your attention. I'm just going to show you where it is and give you 30 seconds of what it actually does, and then we'll continue on. But this is an example of using the Python API to do model calibration with SciPy. So you can find this demo back in the demos folder near where we retrieved our original cell sorting simulation. So back in the main Python installation directory, you can go back to demos and go to Callable CC3D from there. So main installation directory, demos, Callable CC3D, and you'll find a directory just after our cell sort 2D directory that we messed with earlier. You'll find another directory called Chemotaxis Calibrate Demo. And when you go into there, you should find something that looks similar to what we constructed in our first exercise. And then there's a Python script here named Chemotaxis Calibrate. And then there's a directory, Chemotaxis Run Model. And when we go in there, what do you know? There's a CompuCell simulation. And again, I'm not going to dive into this for more than half a minute. But what this Python script does is go all the way down to around line 132. For those of you, say, who took the course last semester or who have worked with SciPy in general, this may look familiar. So this is operating with a SciPy optimize function called minimize scalar. And what this is using is a cost function that is being driven by CompuCell simulations. And the cost function here is designed to find a chemotaxis parameter that minimizes the final distance of a chemotaxing cell and some point in a simulation domain. And the objective here is to calibrate this chemotaxis model parameter to essentially find the parameter that produces a particular speed. Now, when you browse through this, you should find some familiar things with respect to what we discussed today, in particular around line 87. You find that there is an instantiation of some CC3D caller classes. But as you browse around, you should also find lots of other things going on that we didn't have time for today. In particular, things like a CC3D caller worker. This is support for multiprocessing. So executing multi-threaded applications, doing lots of CC3D simulations simultaneously, for those of you who may be interested in, say, HPC deployments. But then also doing a little bit of analysis on results for multiple runs, doing a bit of statistics, and then constructing a cost function that can be used so that CC3D functions as feedback during model calibration. So again, that's in the main installation directory. That's under demos, callable CC3D, chemotaxis calibrate demo. This is not an official assignment unless James determines otherwise. I will say just that for those of you who may be interested in some basic applications, that this is a great place to start. And so my concluding thoughts are mostly just along those lines, that this very much broadens the utility of CompuCell to function in lots of advanced workflows, whether it be model calibration as demonstrated in the chemotaxis calibrate demo, or integrating artificial intelligence to do, say, training for model surrogates. One of the applications that we tend to use this the most in is for HPC deployments. So for using just basic multiprocessing for multi-threaded applications, we can spawn lots of instances of CC3D simulations in an HPC environment. This example, this is from some of our work in viral infection from last year. In particular, there are 640 simulation instances that are represented here. Each of these little lines is a different simulation instance. And this is equivalent to about 160 hours of simulation time if you were to run each of these one after another. Whereas using the CC3D API and just basic integration using Python's multiprocessing package, dumping this on IU's Carbonate reduced this entire simulation time to about 8 hours. So pretty powerful stuff for, you know, just a couple of lines of additional code.
And as I was discussing with James earlier, those type of uses are particularly useful when using CompuCell for research purposes. When we usually use Player and CompuCell Player to build our models, and once we have built our models and we have calibrated them, and we want to do exploration of the model parameters, we stop caring about running those simulations in Player, and we start running them in different settings, and then all we care about are the inputs and outputs of those simulations. And this is one way in which, and a particular powerful way in which this can be done. So now let's come back to implementing the intracellular SBML models. I'm gonna go a little bit fast over the biology because this is some material we have been seeing over the last 3 weeks, and I want to get to the important part where I showed how the code is implemented and how to add steering panels to our model. So the first thing, the basic idea about the delta notch patterning is it's a basic signaling network between cells, and it is what we usually called in cellular regulatory networks as a toggle, which means that in a single cell, the cell can adopt one of two states. It can be high in delta or high in notch, but it cannot be high on both. And what we see in biological systems is that this toggle, this regulatory networks usually leads to this patterning where we get cells that are on and off in a regular pattern, and this is important for different biological processes, especially in developmental processes. And again, the idea in this model is that a cell that is high in, a cell that is high in delta will be, I'm sorry, let me finish this poll here. a cell that is high in notch or it's activated in notch will decrease the delta inside the cell. So a basic model of this idea is encapsulated in this differential equation model. And I'm not gonna go over the full details of the model, but the idea here is that you have a activation of delta. Let's start in notch. You have an activation of notch that is dependent on the average delta of the cell neighbors. So this delta bar refers to the delta signal that is transmitted to the cell by its neighbors. So if the cell has high, the cell neighbors are high in delta, then notch in this particular cell, in the cell I care about, is gonna be activated. And then activation of notch leads to inhibition of delta in that same cell. And that's the basic idea. And that model will lead to, for those of you who are not familiar, for those of you who took the class with us last semester, these functional forms should be familiar to you. This is a Hill function. These are Hill functions, and this is an activating Hill function of delta, and this is a inhibiting Hill function of notch. So delta activates notch, notch inhibits delta inside of a single cell. As I said, this leads to this high-low notch patterning shown in this, and we would expect to get from our simulations when we simulate this in a special context, a patterning that resembles this simulation. This simulation here, or this image here. So what we will do the way of implementing this is by having a anti-money model or an SPML model that corresponds to the delta notch model that I explained earlier, and instantiating this model inside every cell of our simulation. And then once we have instantiated that inside the cell, we have to connect the value of delta average of the model inside a cell with the delta value over the cell neighbors. And then that will create the characteristic delta notch patterning. When we run the simulation, this is the characteristic notch pattern that we should expect to see. Okay, so the first part is to have a SPML model or of anti-money model of the delta notch, SPML or anti-money of the delta notch model that I was explaining earlier. And I was explaining last time that models are usually shared as SPML because that is one of the standards for model sharing. And SPML is a widely used markup language in which models can be shared. The reason we prefer anti-money models the reason we prefer to convert SPML model into anti-money strings is because anti-money strings are more readable from a human perspective in terms of it's easier to understand how the variables interact and what the equations of the model are, what the parameter values are, and so on, by looking at the anti-money rather than opening the SPML. And for translating from SPML to anti-money models, we can use a tellurium method Tellurium is a package that can be used for simulating SBML model and antimony models. And it has a specific method called SBML to antimony that allows you to translate one SBML to antimonies. And when we, the specific SBML model that we are, that contains the delta notch is when you translate it into antimony, this is the resulting antimony model that you get from it. And when you look at the equations, these equations correspond to the equations that we were looking at earlier. Specifically, you can see that you have the delta average activating notch and notch inhibiting delta. And this model is gonna go inside every cell of my CompuCell simulation. So now that we have the model, we can we have to load it into our, going back here, it's because this is our step 0, and this is the idea that James was explaining earlier. It is an excellent idea, and we recommend before loading your SBML or your antimony models into CompuCell to have a package such as Tellurium that allows you to execute this these models, so you have you get some familiarity with the dynamics of the model independent of CompuCell, and then that way you can. For example, I could explore what the state or what the steady state of my system will be, or the fixed points of my system for those who took the class with us last semester will be for different values of the delta average or the delta average parameter. That it's the one that I'm going to be linking with my CompuCell simulation.
Right.
Go ahead.
That's a good example, though, that this particular case you have to think carefully because delta and notch normally exist in opposite cells. So D average is the amount coming from the neighboring cells. So you could think of that as a parameter here, or you could imagine building a model, 2 different antimony models, J1 and J2 and J1 prime and J2 prime, and imagine having 2 cells that are cross-coupled to each other.
All right, and we've done that too last semester, where we created different instances of the antimony models, and then we make them communicate with each other. But in that particular case, every single antimony model, right, is every instance of the automotive model corresponds to a single cell in your will eventually correspond to a single cell in your simulation. But you should start by having a single antimony model that you explore the ranges of these parameters that you're gonna later link to CompuCell properties and outputs. I'm sorry, James, I interrupted you.
No, I was done. I thought I was agreeing with what you were saying, basically. I just wanted to point out that in this particular case, the simple model might be D average as an external control parameter, or you might have 2 cells that couple to each other, but either way, putting it into antimony is not a bad idea at all.
Right. So, but again, the idea here is, the main idea is that it's wise to explore the model as a standalone before implementing into CompuCell. So you have a familiarity of how the parameters of the model control the dynamics when you simulate it. Once you have that sense of what the model does, and this is, again, it's important to point it out, because you don't want to load a model into a cell that you have no idea what it does. But once you have that, once you have that familiarity with your model, then you can load it into the cells of your simulation, into your CompuCell cells. And since we have both the SBML model and the antimony string, there's two ways we can do that. We can load it directly as an SBML model using this. There is a typo here. This should be in the next line, but using this add SBML to cell types method. And then we just have to specify what the file is that contains the SBML model, what the name of that model it's gonna be. And this is important because this is the name that I'm gonna use later to reference the model. What are the cell types that I'm gonna be attaching this model to, and what is the step size? And this is basically the correspondence between a Monte Carlo step and a simulation and a step in the intracellular model. And this is something that also needs calibrating. We usually recommend 1-to-1, and then you can add up parameters, or you can set as the step size as you prefer. That will probably require some exploration as well on your part. But the other option for you to load it is to do it using directly the anti-money string, and you do that by using this syntax: add anti-money string, add anti-money to cell types. And instead of specifying the model file, here I specified what the model string is, and I have defined it previously. And again, I need to specify what the model name is and what are the cell types that are going to be attached in this model to, and what the step size correspondence is. There are other options. I can, rather than loading to cell type, I can load it just on a specific cell. I can load it by cell ID. And there's other options that you can explore, but the general syntax will be the same. It will be add anti-money cell, add anti-money to cell, and then whatever specific cell you want to do to attach that model to. It's important to notice that these two are equivalent, and if you have an anti-money string that correctly encapsulates what you have in the SBML model, it should be equivalent for you to use either of these two. And this is a good way of checking that you have a correct anti-money model that describes that corresponds to the SBML model. Then if we usually do all of this in a star function. So this is the way, or when we create cells, when we instantiate this anti-money models into the cells, and then we usually need to specify what the initial conditions of them in the model are. And in this particular simulation, in the delta notch simulation, the way we initialize the values, the delta and notch values, is that we assign a random number between 0.9 and 1. And that is the value of delta and notch that each of the cell is going to have. And look, this is important. The way I reference the delta value inside each cell, the delta and notch value inside each cell, is that I loop through my cells, my cell list, and once I reference a specific cell, then I grab the SBML that model name DN, which I specified before. And this is what I meant before. This is when I pointed out that was important, because this is the way I write and read and write values from and to the SBML model. So as I said, if you run that model, your cells will run, each of them will run a independent anti-money model, but they won't be connected because we haven't tied any parameter or value of the model to a specific property that you gather from your CompuCell simulation. The easiest way to connect this model, to link each of the intracellular models to a CompuCell simulation, CompuCell property, is to conceptually to iterate over, let's say I picked a specific cell, then I iterate over the neighbors of that cell. I read what the delta value of those neighbors are, and then I average that value over the neighbors. And the specific syntax to implement that idea is here. I iterate over my cells, and then I determine what the neighbors or my cell are, and we've used this syntax before. And we've pointed out that we always have to iterate over, we always have to include this conditional if, because iterating over the neighbors will include medium, but we do not care for medium as a neighbor. And in fact, we don't have a SVML model loaded into the medium, so that would that would return an error. So we have this condition to ensure that we care only about the neighbors that are actually cells. And then what we do here is that we are adding, right? We're adding the delta of all of the neighbors, and then we're averaging over the we're average that quantity over the number of neighbors. So basically now we tied up the delta average inside each of the cells to the delta of their neighbors. And then finally we time step the model. And once we've done that, we've finalized the simulation. So let's run that simulation. I'm gonna stop sharing real quick, and let's run that simulation so we can see what it's doing. So I was mentioning earlier, we have the anti-money string, and I'm gonna upload this code so it's available to you. We have the anti-money string corresponding to the delta notch model. Then in the start function, we're loading the anti-money to each of the cells, and we're initializing the delta and notch values inside of each of those cells. And then in the step function, we're averaging the delta over the cell neighbors. And you can see here in line 45 that we're writing back on to the cell what the delta average is. So now with this specific line 45, I am linking the delta of the neighbors in CompuCell with the delta average in the inside of the cell in the internal cell SML model. And when we run that simulation, what we see is that we get the specific pattern where cells are either high in delta or high in notch, but not both. And then we get this specific patterning that is characteristic of the delta notch model. Okay. And then we can do more complicated stuff. We could, for example, start with have a different initial condition, and I'm just gonna show it up, and I'm gonna show the code. But we could have something more complicated where we start with a single cell, and then based on what their value of notch is, we can have the cell growth and divide, or if the cell is high in delta, then have the cell stays the same size. And then we can explore what happens to the patterning when we combine cell growth with the delta notch. And I think, James, you can correct me, I think actually notch is a promoter of cell division. I'm not sure I read that somewhere, but.
Yes, that's right. It can act as a transcription factor.
Right. So in this particular case, you guys see that the cells that are notch are the ones that are growing. The cells that are delta remain the same size, and then that's why you see the cell division occurring just on the no, I actually they occur on all of them. I need to make sure that I updated the code, but in this case I made all the cells grow, but I could add easier. I can easily add a condition where only the cells that are high on notch are the ones growing and dividing. So that was just to show how I could link delta notch to growth, but I wanted to show how to add steering panel because this is going to be important for your simulation in the next couple of minutes. So the way of doing this is by adding a specific function inside a CompuCell steppable that we called add steering panel function. So we're going to go back to our delta notch model. And we're going to add, by the way, this is the first time I'm trying this myself. So we're going to add the add steering panel function. And this function takes self as an input. And then let me see if I can just move this way. And then we want to now inside this function, we want to specify what are the parameters that we're going to be adding to that steering panel. And then let's just add for now as an example, because we know that lambda volume is going to make the cell more or less motile. Let's just add lambda volume. So self that add steering param. And then the name of the parameter that we're going to be changing. Let's call it lambda volume, just not to confuse with the actual property of the cell. And then we have to specify what the values of what the values are. Let's say min val min val 4, min val, let's set it to 0, and max val or to 1 because we don't want our cells to disappear. Let's make it 20. And then a widget name, let's call it slider. Right. So now that we have added, val is gonna be the default value, and that's gonna let's set it to 2. Now that we have added that steering panel, let's define our function that is gonna allow us to update the values as we change the steering panel. And that's called process steering panel data. and it also takes self as a input. And then here, what we will do is that we have to, because every time we update the value, we need to basically iterate over our cell list and then update the value of the lambda volume. So what we will do is that we will iterate over our cell list, and then we will update the cell's lambda volume to be equal to this lambda volume that we have specified in the staging panel. Okay, let's just make sure that right. So let's see whether this works. There you go. So now we have a staging panel. Okay. Do I have to pass something else? Oh, I'm sorry. I need to get the value. So let's call it here lambda volume self that get staging parameter, and then it's called lambda volume. Okay. And then I can so this is supposed to be updating the lambda volume of my cells. A way of checking that is to print the cell lambda volume to make sure that we're actually, and this is why the printing statements are included in that simulation, in that example, because that's a way of checking that your lambda volume is. So this is not being called, or is this being called just when I change it? Yeah, so it did change. So maybe let's maybe increase it. Let's increase the range in which we want to allow the lambda volume to change. Let's make it 200. Let me stop my simulation again and launch it. Yeah, though doesn't seem to be doing much, but it's definitely, oh yeah. There you go. It does seem to take a little bit. Wait. Right, there you go. So you can see how I'm updating the lambda volume. And as I update the lambda volume, the fluctuations of my cells change. And this is just a very simple way of adding a staging panel that allows you to control a parameter in the simulation. There you go. I did not launch the poll. Were people able to follow through with at least the staging panel? It's not very complicated, really. See how I'm gonna recap in the last 2 minutes. So we add this add staging panel method inside of a stepable. And then we specified a name for a name that is the name that it's going to appear here. You want it to be descriptive of the parameter that you're going to be changing, the range, and the values. And then there's other options. There's, besides the slider that I showed, there's also different options. There's a pull down menu, but I'm not exactly sure what the names are. I'm going to consult with Giuliano. And then after we have specified we have initialized the steering panel, then we need this process steering panel data function that basically allows us to update the parameter in the simulation as I manipulate the value in the steering panel. And I need to get the value of that from the steering panel and then pass it on to whatever simulation input I am interested in changing. And I'm gonna stop there because it's 8, but I hope that was informative. So James, I'm gonna pass it on to you now.
Okay, well, I hope people can play with the steering panels a little bit, because the key idea, which we saw already if people who took it last semester and people who've worked with Jupyter Notebooks, is that the steering panel is asynchronous. So whenever you move the slider in the steering panel, or whenever you do the pull down or the type in the steering panel, that will then call the process steering panel data routine. And so your program will run continuously while waiting for those inputs. If you want to actually wait and not have the program run, then you have to create an empty loop which sits there until the steering panel is used. And that's something that you could do if you need to. So for example, suppose that you had a pull down menu which said run one of 3 simulations. You could put that in the start function and then have the program sit there and do nothing until the user selected one of those 3 things, and then start the simulation. Again, from the point of view of running it on your desktop, that's probably not very useful, but from the point of view of a simulation that you're going to run on NanoHub where the user doesn't have access to the code, that can be pretty useful. Since we don't have the ability to display to the console and ask the user to say type 1, 2, 3, or 4, you could use the steering console to do that. So we could probably share that little cancer demo I wrote, which runs that way, where you select a variety of different possibilities with the pull down menu. So, for example, if you wanted to write a simulation that might do the, say, Colli's simulation for dictyostelium, you might want to have. I mean, the one thing you can't do with the steering panel is that the XML is always going to execute before the steering panel. And so anything that's in the XML, you're stuck with. But anything that you do in Python, for example, if you created a different initial condition of pattern of cells laid out in different ways, you could have the steering panel select how to lay them out, anything that you would do in the start function. And again, if people want to talk about that one-on-one as it relates to their particular simulations, I'm happy to